Now we will discuss the enzymes in the biomolecules. Enzymes, almost all enzymes are proteins except the ribozymes. Ribozymes are catalytic RNAs that occur in nucleus. Active site is the pocket of enzyme into which a substrate fits. Substrate is a reactant which is converted into product. Number of substrate molecules converted into product molecules per unit time is called turnover number. Carbonic anhydrase is the fastest enzyme with the turnover number 6 lakh per second. In absence of enzyme, only 200 molecules of H2CO3 are formed in 1 hour. Hence, the enzyme accelerates the reaction rate by about 10 million times. During reaction, substrate passes through the intermediate transition state. Transition state is high energy state. The difference in energy of substrate and that of transition state is called activation energy. Enzymes increases the rate of reaction and folded by decreasing the activation energy. This makes transformation of a subunits to products. The products is what are the factors affecting enzyme activity? First point, temperature. The temperature at which enzymes are in the highest activity is called optimum temperature. Low temperature preserves the enzyme to temporarily inactive state, whereas the high temperature destroys enzymatic activity because proteins are denatured by heat. Denaturation is loss of tertiary structure. In human, 37 degrees centigrade is the optimum temperature for enzyme activity. Second factor is the pH. The pH at which a particular enzyme shows highest activity is called its optimum pH. Alternation in pH affects tertiary structure of enzyme and hence it is activity. Example, the peptides act in acidic pH provided by HCl in stomach, whereas the trypsin works well in basic pH. Next factor, concentration of substrate. With increase in substrate concentration, the rate of enzymatic reaction rises at first. The reaction ultimately reaches a maximum velocity which does not exceed further with increasing concentration of substrate. This is because the enzyme molecules are fewer than substrate molecules, and after saturation of these molecules, there are not free enzyme molecules to bind with additional substrate molecules. Next factor is the competitive inhibition. When the inhibitor closely resembles the substrate in its molecular structure and inhibits activity of enzyme, it is called competitive inhibitor. Due to its close structure, similarly with the substrate, it competes for the active site of enzyme. Consequently, substrate cannot bind and enzyme action declines. Example, inhibition of succinic deoxyhydrogenase in the Krebs cycle by the malnate. Competitive inhibition is a reversible inhibition as it can be overcome by increasing the concentration of substrate. Competitive inhibition is used in control of bacterial pathogens. Example, Sulfur drugs inhibit synthesis of folic acid in bacteria by competing with its precursor amino acid. Performed folic acid is obtained by animal cells. Therefore, sulfur drugs do not burn them. Non-competitive inhibition. It is irreversible inhibition of enzyme activity by a substance that has been structurally similar with the substrate. Thus, the chemical does not competitive with the substrate in the active site. Example, cyanide kills animals by inhibiting enzyme oxidase of electron transport chain and therefore inhibition synthesis of the ATP. Now the important point, classification of the enzymes. Oxidoreductase, and the first enzyme is oxidoreductase and dehydrogenase. These catalytic transfer of the electron of hydrogen atom. Example, cytochrome. Second, it is a transferase, catalytic transfer of a group from one substrate to another. Third point, hydrolase. These enzymes break up complex molecules into simpler ones with the help of water. The reaction is called hydrolysis. Hydro means water, lysis means breaks. Not all digestive enzymes are hydrolysis. Fourth enzyme is lyases, breaking a molecule without hydrolysis. This reaction may lead double bond in product. Fifth point is ligase. These enzymes catalyze linking of two smaller molecules to form a larger molecule. Example, DNA ligase joins two small DNA fragments to form a larger DNA. Sixth point, isomerase. These are the enzymes which catalyze inner conversion of the optical, geometrical or positional isomers. Next, cofactors. Non-protein components which are bound to enzyme to make them catalytically active. In these enzymes, protein portion of enzyme is called apoenzyme. Prosthetic groups are the organic compounds that are tightly bound to apoenzyme. Example, heme. Coenzymes are the organic compounds, but they are associated with the apoenzymes only during catalytic reaction. Many vitamins form essential components of some coenzymes. This is why deficiency of vitamin in body leads to the appearance of the symptoms. Thank you.